All right, let's jump right in. Today, we are diving deep into a really revolutionary field in medicine. We're going to talk about the science of how our cells actually talk to each other, and more importantly, how we might be able to tap into that secret language to heal our bodies from the inside out. So, what if your cells could send little text messages to each other to fix things when they go wrong? I know it sounds like something straight out of sci-fi, but it's literally happening inside your body right now. Your cells are constantly firing off these tiny packages of information back and forth, coordinating everything from just your daily upkeep to full-on emergency repairs. And these are what we're talking about today, nature's tiniest messengers. Okay, here's what we've got on deck. First, we'll cover what these tiny messengers are. Then we'll get into the cell-free advantage, talk about the challenges of producing them, see how they can unlock regeneration, and finally, look at their journey from the lab to, well, real life.
So, Section 1, Nature's Tiny Messengers. Let's really get into the science of how our cells communicate.
All right, so at the very center of this whole communication network are things called extracellular vesicles, or EVs for short. The best way to think about them is like tiny cellular postmen. Now, one of the most important types of these, and the one we're focusing on, is called an exosome. Its entire job is to carry really critical cargo, things like proteins and RNA, to other cells, basically delivering a super specific set of instructions for healing and repair. So what's actually inside these little delivery packages? Well, if you could open one up, you'd find a bunch of important stuff. You've got proteins, like growth factors. You've got lipids, which create that protective outer bubble. And maybe most importantly, you have nucleic acids, like mRNA. And here's the key. These aren't just random bits and pieces. They are specific, targeted biological instructions that can literally change how the recipient cell behaves. Okay, Section 2, the cell-free advantage. Now, this is where things get really interesting, because it turns out just using the message from the cell might actually be a whole lot smarter and safer than using the entire cell itself. You know, for years, stem cell therapy has been this huge beacon of hope in medicine, right? But it's got some real challenges. When you infuse whole cells, there's always a risk of your immune system rejecting them, or even the scary possibility of them transforming into something you don't want, like a tumor. Plus, storing them is a nightmare. But this is where exosomes therapy just flips the script. Because it's cell-free, you're only using those little messenger bubbles, not the whole cell, the risk of rejection or transformation just plummets. They're also way more stable and easier to sterilize. It's a huge potential leap forward. And look, this isn't just us talking. The science backs it up. This quote from a major review in pharmacology says it all. For some therapies, these vesicles might actually be superior to using whole cells. They're easier to work with, they're more biocompatible, and they have fewer risks. I mean, think about that. It means you could get all the incredible regenerative power of stem cells without actually needing the cells themselves. That's a game changer. Okay, so if these exosomes are so amazing, you're probably wondering, why aren't they being used everywhere already? And that brings us right to Section 3, the production challenge. Because as you can imagine, making and handling these things is, well, it's not simple. So how do you even make these things on a large scale? Well, here's a pretty typical approach. It starts with growing stem cells, often taken from fat tissue, in a special culture. Then comes the conditioning step, where you basically coax the cells into releasing all those good exosomes into the liquid around them. After that, you have to very carefully filter everything to get rid of the cells and any junk, leaving you with this valuable, exosome-rich conditioned media. And of course, the final step is intense testing and validation to make sure it's sterile, safe, and has the right concentration of exosomes. All right, so you've gone through all that work. You've made your batch of pure exosomes. But now you have a new problem. How in the world do you keep these super delicate little bubbles stable so they can actually be used as a medicine later on? Well, the obvious first thought is just to freeze them, right? But that has a huge problem called cryo-injury. Think of it like this. As the water freezes, sharp little ice crystals form, and they can act like tiny daggers, piercing and rupturing the delicate exosome bubbles. Not good. So, scientists came up with a much smarter method, lyophilization, which is just a fancy word for freeze-drying. It's a process that gently removes the water at a really low temperature, turning the whole thing into a stable powder. You can store it, ship it, and then just add water later to bring it back to life. No daggers involved. But even that amazing freeze-drying process needs a little extra help. To really make sure the exosomes survive, scientists add what are called protective agents, or excipients. These are things you might even recognize, like certain sugars, polymers, and amino acids. What they do is form a tiny, glassy protective shell around each and every exosome. It's like bubble wrapping them on a microscopic level to keep them safe and sound. And this step is absolutely critical if you want to create a stable, off-the-shelf product.
So, scientists are figuring out how to make them and how to keep them stable. Now we get to the really exciting part, the payoff. Section 4 is all about unlocking regeneration. Let's look at what this technology could actually do. One of the biggest areas where exosomes are showing incredible promise is in skin and wound healing. It seems like they can just supercharge the whole repair process. They tell skin cells to multiply, and they ramp up collagen production. What does that mean for us? Potentially, it means wounds that heal way faster and with much less scarring. That's a huge deal. And it's not just about skin. They're also looking incredible for bone repair. And they do it in a really clever way with this kind of dual action mechanism. On one hand, they help block the cells that are responsible for breaking down old bone. And on the other hand, they protect the cells that are busy building new bone. It's this 1-2 punch that makes them so perfect for helping fractures and other bone injuries heal up faster.
Here's another really cool application, this time in cosmetic and reconstructive surgery. You know, fat grafting is a really common procedure, but it has one major flaw. A lot of the fat that's transferred, well, it just doesn't survive. It dies off. But get this. Researchers are finding that if you add exosomes to the mix, you can dramatically improve how much of that graft survives and keeps its volume. And the secret sauce seems to be their ability to kickstart the growth of new blood vessels, basically giving that new tissue the vital lifeline it needs to live and thrive.
So as you can see, the potential here is just massive. We're talking about everything from skin regeneration for anti-aging and scars, to helping with bone and cartilage issues like osteoarthritis, to calming down inflammatory conditions like dermatitis, and of course, what we just talked about, tissue augmentation with fat grafting. It's incredibly broad.
Which, of course, brings us to our final section, from lab to life. What's it actually going to take to get this incredible science out of the research lab and into the doctor's office? So getting this to patients isn't a walk in the park. There are some major hurdles to clear. First, quality control has to be absolutely airtight. Every single batch has to be consistent and powerful. Then, you've got to figure out how to manufacture these at a massive scale. After that comes the big one. Large, expensive clinical trials to prove, without a doubt, that they're safe and that they actually work in humans. And finally, you have to navigate the very complex, very long road of regulatory approval.
So yeah, the path is definitely challenging, but at least it's a well-defined one. It always starts with the basic research and discovery. Then you move into developing a manufacturing process that can be scaled up. From there, it's preclinical testing, usually in animal models to check for safety and see if it works. Only after all that can you move into the multi-phase human clinical trials. It's a long, long road, for sure, but it's the one you have to travel for any truly revolutionary medicine.
So let's wrap this up. We've seen how these incredible exosomes act like nature's own postal service. We've talked about their huge advantages over older cell therapies, and we've laid out the long road to actually getting them into the clinic. The journey is definitely complex, no doubt about it, but the potential is just undeniable. Which leaves us with one last massive question. Could these tiny, tiny messengers end up being one of the biggest leaps forward in modern medicine? Only time and a whole lot more research will give us the answer.